All right, you made it. Last of the energy slides. This last presentation is the Calvin cycle, but then we're also going to cover some alternate pathways that photosynthesis can take. Okay. The Calvin cycle has a couple of AKAs. The light reactions, the light-dependent reactions, that's pretty clear, right? They require light to go on. The Calvin cycle was named after the guy who discovered it, and that was Melvin Calvin, and yes, his name was Melvin Calvin, yes. But also, the Calvin cycle is often referred to as the light-independent reactions, which is fairly clear too. These can occur independently of light. Sometimes they're just kind of lazily called the dark reactions as well. They don't have to take place in the light. It's just that they can take place whether there's light or not. So they're independent of light, okay? Now, the first part of photosynthesis was about generating energy, right? Generating chemical energy. And we got a couple things that we needed from that. We got ATP and we got NADPH, and now we're going to use those things to create sugar, right? And this is a cycle. And one of the things we kind of glossed over in the last video or not the last video, but the video on the Krebs cycle, I just want to highlight here. Cycles always start with kind of a reaction phase, right? And then they go into a phase where you have to regenerate your starting molecules. Since they're a cycle, you need to regenerate that starting molecule so that the next molecule can come in, that comes in has something to bond to. And so the Calvin cycle actually is split up into 3 phases. There is a carbon fixation phase where carbon dioxide is going to come in. Okay, so from the equation for photosynthesis, CO2, I don't know why this is erasing here, CO2 is going to come in to the equation or to the reaction here. That's coming into the carbon fixation phase. Then you're going to get reduction. So that's where we're going to actually remove phosphates from ATP and remove electrons from NADPH. And we're going to reduce some molecules in the Calvin cycle to produce something called G3P. This is also sometimes referred to as PGAL. More modernly, it's G3P. I think PGAL is the old name. But this is a sugar molecule that will eventually be converted to glucose. So think of it as a half sugar. So every time you turn the Calvin cycle around twice, you get 2 half sugars that can be bonded together to make a glucose. And then the third step is going to be where we actually regenerate the RUBP, which is the first molecule that we start with. RUBP is ribulose bisphosphate. And RUBP is our starting molecule, so we're going to regenerate at the end and allow that cycle to start over. So the Calvin cycle, the first thing remember is the where. We said in the last video, this is the stroma of the chloroplast. And we said carbon dioxide is going in, right? But so is ATP from the first step from the light reactions and NADPH. Okay. Now, I'm sure you can guess that the ATP is going to come out as ADP, right? Because we're going to use the energy from ATP. We've already said that. And we've said we're going to strip the electrons here and do some reduction to make the G3P. So we're going to go back to NADP plus here. Okay. So let's look at that and then we'll come back and kind of finish out what happens here. Okay. So what you see here is the RUBP. This is our starting point right here. RUBP bonds to carbon dioxide. That's why this is the fixation phase. And there is an enzyme that you need to know about here that I'm not sure. I've seen it pop up in College Board questions. The enzyme is called Rubisco. And of course, it has a longer, more complicated biochemical name than just Rubisco, but you need to know that name. Rubisco is the enzyme that's responsible for fixing the carbon dioxide and attaching it to the RUBP to create this phosphoglycerate. You don't need to know the name, phosphoglycerate, but RUBP is something you should be familiar with as the starting point, and Rubisco you should know is the enzyme that's going to drag that carbon in and attach it to the Rubisco. All right, once that happens, you're going to have a number of intermediates here that we're not showing. But notice we're going to take ATP here, 6 ATP. We're going to take 6 NADPHs. We're going to release the energy from ATP, and we're going to release the electrons from NADPH. And we're going to end up with this G3P. And again, this is a half sugar. That's a half a glucose molecule. So if we turn this Calvin cycle around twice, we produce one glucose molecule. Then we go to this regeneration phase where we're going to use some more ATP, another 3 ATP molecules to regenerate our starting molecule, our RUBP. Okay, so that's the Calvin cycle. The only thing we haven't got yet in our chart is that this CO2 is going to get incorporated into the G3P. Okay, so the CO2 is going to come out as a component or a part of G3P. And the point is we made sugar, which is like why we do photosynthesis, right? We made the sugar. Yay. Okay, we did what we set out to do. We've made sugar here. So that's what's happening in those steps in the Calvin cycle. Now, there are some other ways that plants have to fix carbon. Okay, we're talking about plants here, and plants live in some pretty crappy areas sometimes, right? Sometimes they live where there's constant wind blowing, like at the beach. If you've got an oleander plant, for example, or you've got cactuses that live in the desert, and they're going to have some problems, right? Think back to that leaf anatomy, right? The leaf is designed to bring together all of the parts of photosynthesis into one spot so that we can actually perform photosynthesis. But that relies on being able to bring water up from the roots, right? It relies on light being able to strike the leaf. It also relies, though, on the stomates being able to be open to let air in because air contains carbon dioxide, right? So what we've been discussing here, this photosynthesis we've been doing is called C3 photosynthesis, okay? And that's because RUBP will, in the first stages when carbon is fixed, break into 3 carbon compounds made of 3 carbons. The problem here is that if you have a C3 plant in a hot place, they're going to have to close their stomates during the day to prevent water loss. And during the day, when the sun is shining, that's when plants don't want to close their stomates because they need to bring in carbon dioxide. While the sun's shining, the products of the light reaction start to build up. You get a bunch of ATP and NADPH building up. The plant wants to bring carbon in and start fixing it through the Calvin cycle to create sugars and start building up sugars. Because plants like to store sugar too, right? They like to create a bunch of sugars. They'll string them together into starches and shove them down into the root and store them there. Sometimes they store those sugars as starches in the stems, in the leaves, and so on, right? So plants, in order to adapt, have to come up with alternate ways for being able to get that carbon dioxide. So there are 2 alternate pathways for plants that consistently live in hot environments. The first is the C4 pathway. C4 plants have what are called, well, all plants have what are called bundle sheath cells that surround their veins. So when you looked at that leaf diagram in the video before, you saw the vein with its phloem and xylem, and then surrounding the vein was a layer of cells known as the bundle sheath cells. And the Calvin cycle only occurs in those bundle sheath cells for C4 plants. But carbon is actually fixed in the mesophyll in the form of a 4-carbon compound. Okay, this is why this is called a C4 plant. There's an enzyme PEP carboxylase that actually helps to fix carbon in those mesophyll cells. So like in the spongy cells surrounding the bundle sheath cells. And so essentially this is a space separated, right? You're separating fixing of carbon and using carbon for the Calvin cycle into 2 different areas so that catch as catch can, whenever those stomates can be open, now all of those mesophils are brought into action to fix carbon into a 4-carbon compound. And then when the plant can do the Calvin cycle, when it's got enough ATP and NADPH to do the Calvin cycle, then those mesophil cells will feed that 4-carbon compound, that fixed carbon, into the bundle sheath cells through their plasmodesmata, and photosynthesis can proceed as normal. Okay, so we have fixation and use occurring in 2 different places. Okay, so they're isolated by place in C4 plants. Cam plants do this a little differently. Cam plants do fixation in the same cells where they do use, where they do the Calvin cycle. But what they do is build up something called crassulacean acid in those cells, in the mesophil cells where the Calvin cycle is taking place. Okay, so these plants will only open their stomates at night, and as those stomates are open at night, they collect carbon dioxide and store it into crassulacean acid. And this is called crassulacean acid metabolism, or CAM for short, right? This is why this is CAM photosynthesis. So now in the same cells where the carbon cycle is going to take place, you're storing up a bunch of this CAM, or this crassulacean acid. And then during the day, when the stomates have to be closed, you already have an amount of fixed carbon already there. The plant can then take that crassulacean acid, break it down, and then use the carbon dioxide to create sugars in the Calvin cycle. Okay, so 2 different pathways for that. So there's the leaf surfaces, there's the stoma. This is an open stomate. That one is open. These are the little guard cells that surround it. And remember that these stomates have to be open to let carbon dioxide in. Now, water vapor can escape. That's transpiration, right? But carbon dioxide can only get in if those stomates are open. All right, so here is a C4 plant. Okay, so here's the vascular tissue right in here, and then this layer of cells right here is the bundle sheath right here. Whoops, sorry. This is the bundle sheath right here. These are bundle sheath cells right there. Okay, and these cells in here are where the Calvin cycle is taking place. Okay, so this is that layer. And again, you're separating, this is C4, so carbon dioxide is coming in whenever the stomates are able to be opened. And it's being fixed in this C4 compound, right, in the mesophyll cells. And then it's being passed when needed. The carbon dioxide is stripped off of it, and added to Rubisco and so on to do the Calvin cycle. What remains, of course, is a C3 compound that can be attached to another carbon dioxide whenever the stomates can be open. Okay, so that's C4. And then this is showing them side by side. Okay, so again, here's C4, here's creating this cellular isolation of carbon fixation and the Calvin cycle. Here, what you have is temporal isolation. You're fixing carbon in the same cell in CAM, but you're fixing carbon at a different time from when you're actually going to be using it. Okay, so this little dark panel over this is indicating you're actually fixing carbon at night, and then you're actually using it during the day. And those are the alternate pathways of how photosynthesis can take place.